When geneticists at the University of Adelaide extracted DNA from 92 pre-Columbian mummies and skeletons scattered across the Americas, some pulled from caves in Mexico, others from burial sites stretching all the way down to Chile, they expected a pattern, a thread, some familiar genetic signature still carried by living indigenous people today. They ran the sequences, they compared them against every modern database available, and what they got back was silence. Not a single match. 92 ancient humans spanning 8,000 years, and not one of them had a genetic lineage that exists in any living person on Earth. That was 2016, and it cracked open a door that researchers in Mexico had been pushing against for over a decade, because some of the strangest, most confounding DNA results on the planet had not come from Egypt or Siberia or the Sahara. They had come from underneath Mexican soil. This story starts in the dark, literally. In 2007, a team of scientific cave divers led by Alberto Nava was mapping a massive underground cave network called Sac Actun on the eastern edge of the Yucatan Peninsula. They had been swimming through submerged tunnels for hundreds of feet when they reached a void so large their dive lights could not reach the other side. They named it Hoyo Negro, which means black hole. The chamber sat more than 130 feet below sea level, a bell-shaped space roughly the size of a professional basketball arena, buried deep inside the earth. And scattered across its floor were bones. Many bones. Giant ground sloths, saber-toothed cats, elephant-like creatures called gomphotheres. Animals that had not walked the earth in 13,000 years. Then the divers found something else. A human skull, resting upside down on a rocky ledge, surrounded by other skeletal remains, small, delicate. A girl, maybe 15 or 16 years old. They called her Naya, after water nymphs in Greek mythology. What the team did not know yet was that they were looking at the oldest, most complete, genetically intact human skeleton ever found in the New World. Getting to Naya was not simple. The dive required climbing down a 30-foot ladder into a nearby sinkhole, swimming along a 200-foot tunnel through total darkness, and then making a final 100-foot descent into the chamber itself. The pit was 61 meters in diameter, so massive that it swallowed all visible light. Every expedition was a logistical challenge and a physical risk. It took years to organize a proper scientific effort. Pilar Luna Ereguerina, head of underwater archaeology for Mexico's National Institute of Anthropology and History, coordinated the project. Unauthorized divers had already entered the cave, moving some bones and damaging others. The clock was ticking. Researchers used a combination of radiocarbon dating on tooth enamel, uranium-thorium dating on calcite crystals growing on the bones, and sea level rise data to triangulate Naya's age. The results placed her between 12,000 and 13,000 years old. She had fallen into the pit during the last ice age, when the cave system was dry and the water table sat far below. Her pelvis was broken. She likely died on impact. Then the cave flooded around her, sealing her in. Here is where it gets complicated. James Chatters, the lead researcher on the study and an expert on Paleo-American morphology, looked at Naya's skull and saw something he'd seen before. Her face was narrow. Her forehead was low and prominent. Her eyes were wide-set and her jaw jutted forward. It was almost the opposite of what modern Native Americans look like, he said. Her skull fit a pattern known from the very oldest human skeletons in the Americas, remains that resemble ancient populations from Southeast Asia or the South Pacific more than the people living in Mexico or Peru today. For decades, this mismatch had fueled a fierce debate. Were the first Americans actually two separate groups, arriving at different times from different parts of the world, or was something else going on? Chatters needed DNA. 3 independent labs analyzed mitochondrial DNA extracted from Naya's wisdom tooth. Brian Kemp at Washington State University, Deborah Bolnick at the University of Texas at Austin, and Ripan Malhi at the University of Illinois all ran separate tests. All 3 came back with the same result. Naya carried haplogroup D1, a genetic signature that developed in Beringia, the landmass that once connected Siberia and Alaska, and that occurs only in the Americas. It is a founding lineage of modern Native Americans. Her face said she was from a different world. Her genes said she belonged to this one. This was the first time anyone had been able to match Paleo-American skull characteristics with DNA from one of America's founding lineages in a single, well-dated individual. The finding was enormous. Yemaine Asmarom, the University of New Mexico geochemist who helped date the remains, compared Naya's significance to that of Lucy, the 3.2 million-year-old human ancestor found in Ethiopia. The years of methodological refinement in his radiogenic isotope laboratory had paid dividends. "We are gratified," he said, "by the fact that we played a crucial role in helping answer one of the outstanding questions in anthropology." The study, published in Science in 2014, argued that Paleo-Americans and modern Native Americans were not separate populations at all. They came from the same founding group. The physical differences between Naya's skull and modern indigenous faces were not evidence of different origins. They were evidence of evolution happening in place, thousands of years of change within a single lineage after it left Beringia and spread south. Deborah Bolnick put it simply: the physical differences between Paleo-Americans and Native Americans today are more likely due to changes that occurred in Beringia and the Americas over the last 9,000 years. But if Naya's DNA connected her to living people, why does the title of the story say otherwise? Because Naya was one individual. When scientists zoomed out and tested dozens more, the picture changed dramatically. 2 years after the Naya study, Bastian Yamas and his team at the Australian Centre for Ancient DNA, working with Lars Fehren-Schmitz at the University of California, Santa Cruz, David Reich at Harvard, and Wolfgang Hock at the Max Planck Institute, published a study that shook the field. They had sequenced whole mitochondrial genomes from 92 pre-Columbian skeletons and mummies. The samples came from major population centers along the Western Americas, from Mexico down through Peru and Chile, and they dated from 8,600 to 500 years ago. Not one of those ancient mitochondrial lineages showed up in any modern genetic database. Not one. Let that sit for a second. 92 people. 8,000 years of history, and their maternal genetic lines are gone. Completely. Lamas described the finding as completely unexpected. His team ran demographic simulations to figure out what kind of historical scenario could produce such total erasure. Simple population bottlenecks did not explain it. Geographic separation alone did not explain it. The only model that fit the data was one where populations became geographically isolated from each other shortly after initial colonization, stayed that way for thousands of years, and then experienced rapid catastrophic extinction following European contact. The separation between ancient and modern lineages appeared to have been established as early as 9,000 years ago, Yamas noted. Which means the genetic diversity that existed in Mexico and across the Americas before Columbus was not just different from what exists today. It was a different world. To understand how that is even possible, you need to go back further. Back to the bridge. Sometime around 25,000 years ago, a small group of people from northeastern Asia split off from the rest of the continent's population. They moved north and east, eventually reaching Beringia, a vast, cold, treeless expanse connecting Siberia and Alaska, and they got stuck there. Ice sheets blocked their path south, and resources were limited. The population stayed small, maybe a few thousand people, isolated from the rest of humanity. Lars Fehren-Schmitz, one of the co-leads on the 2016 study, described the conditions bluntly. It was at the peak of the last ice age, when cold deserts and ice sheets blocked human movement, and limited resources would have constrained population size. This long isolation of a small group of people brewed the unique genetic diversity observed in the early Americans. For somewhere between 2,400 and 9,000 years, depending on the model, this tiny group lived in genetic isolation, developing the unique DNA signatures that would become the founding lineages of every indigenous population in the Americas. Haplogroups A2, B2, C1, D1, D4H3A. These are the branches on the tree. When the Lamas team built a Bayesian coalescent model using their ancient mitochondrial data, they could actually see the moment those lineages burst outward. Around 16,000 years ago, the lineage diversification rate spikes. The population undergoes a 60-fold increase over just a few thousand years. The ice was retreating, and the descendants of that Beringian population moved south fast, spreading along the Pacific coast and into the heart of two continents. But here's the thing about small founding populations that expand rapidly and then fragment. Genetic diversity becomes patchy. Isolated groups develop their own unique variants. And if something catastrophic happens to those groups later, entire branches of the genetic tree can simply vanish. That is exactly what the evidence from Mexico shows. Consider the Paracues. On the southern tip of Baja California, this group lived for thousands of years in near total isolation. When Spanish missionaries first encountered them in the early 1700s, they noticed something unusual. The Paracues spoke a language completely unlike their neighbors. Their skulls were long and narrow, a shape called hyperdolichocephalic, the opposite of the broader, rounder skulls found in most Native American populations. Their archaeological record in the Cape region stretches back at least 12,000 years. Sylvia Gonzalez, a geoarchaeologist from Liverpool John Moores University, studied their remains and argued that the Paracues resembled the oldest known human skeletons in the Americas, including Pinon woman from the basin of Mexico, dated to over 12,750 years ago, and Kennewick man from Washington state. DNA analysis deepened the mystery. The Paracues did not cluster neatly with other Native American groups. A 2015 genome-wide study by Raghavan and colleagues tested whether they might share ancestry with Australo-Melanesian populations, as their skull shape suggested. The answer was no. But the study also confirmed that their genetic profile was deeply divergent from most other indigenous Americans. They were not from Australia. They were not from a second migration. They were something else, an ancient branch of the American founding population that had drifted so far genetically that they no longer resembled their continental relatives. The Paracues did not survive European contact. Disease, warfare, and forced missionization destroyed them. In 1734, they launched a full-scale revolt against the Jesuit missions, the most serious challenge the order ever faced in Baja California. 2 missionaries were killed. For 2 years, the Spanish lost control of the entire Cape region, but the Paracues paid a catastrophic price. Combat deaths compounded the devastation already wrought by European diseases. By 1768, when the Spanish crown expelled the Jesuits from Baja California, the Paracues were culturally and linguistically extinct. Their genes may persist in trace amounts in local mestizo populations, but their lineage as a distinct people is gone. What remains are bones, artifacts, a handful of recorded words, and cave paintings in the desert that some researchers have compared to Aboriginal art from northern Australia. Remarkably, their genetic fingerprint has surfaced in unexpected places. A 2023 study published in Nature by Nakatska and colleagues, working with David Reika's lab at Harvard, detected a Paracues-related ancestry signal in a 5,200-year-old individual from Central California. The finding raised the possibility that Paracues-related populations were once far more widespread than their final geographic range in Baja suggested, and that their distinctive ancestry decreased over time as other groups expanded. And they were not the only ancient Mexicans carrying lost genetic signatures. In the limestone caves of Tabasco in southern Mexico, a 2024 study led by Maria Teresa Navarro Romero at Cinvestav and Maria de Lourdes Munoz analyzed human remains from Puyil Cave, discovered in 2007 inside San Felipe Mountain near Puxcatan. The skulls in the cave showed artificial cranial deformation, a practice the researchers traced back to the Middle Archaic period, far earlier than previously assumed. Radiocarbon dating placed the individuals across a vast time span, from the Archaic through the Classic period. Their mitochondrial DNA revealed 5 lineages: A, A2, C1, C1, C, and D4. While some showed affinity to modern Maya populations from Mexico and Guatemala, the network analysis also revealed connections to people as far away as Bolivia, Brazil, the Dominican Republic, and East Asia. The cave itself held striking evidence of ritual use: greenstone axes, cinnabar, flint knives, and pectorals placed alongside the dead. These were not ordinary burials. They were deliberate ceremonial acts, suggesting that whoever used Puyil Cave held deep spiritual connections to the land. And their genetic diversity, spanning thousands of years in a single location, captured the kind of variation that would later be swept away. Antonio Salas, at the University of Santiago de Compostela, studied the mummy of a 7-year-old Inca boy found frozen on Argentina's Aconcagua mountain in 1985. Hikers had stumbled across his body at extreme altitude, and archaeologists determined he had been sacrificed around 500 years ago in a ritual called capacocha, where children were taken to mountain peaks and left to die of exposure or killed outright. This boy had likely been executed with a blow to the head and was naturally mummified by the extreme cold. Salas extracted the boy's complete mitochondrial genome from one of his lungs, choosing an internal organ to minimize contamination risk. He even genotyped every member of his own research team to make sure none of their DNA had leaked into the sample. The sequence was unlike anything in any database. It belonged to a newly identified group called C1bi, a sublineage that probably arose around 14,000 years ago in the Andes. When Salas searched every available genetic record, he found only 4 other people on Earth who carried it, 3 modern individuals from Peru and Bolivia, and 1 ancient sample from the Wari Empire, which predated the Inca. Andres Moreno Estrada, a population geneticist at Mexico's National Laboratory of Genomics for Biodiversity in Irapuato, pointed out the statistical implication. If you randomly sample just 1 or 2 individuals from a population and find a rare lineage, chances are that lineage was not rare at the time. It was probably common. What happened was that the population carrying it was destroyed. Up to 90% of Native Americans died within the first century of European contact. Bastian Lamas was not surprised by the genetic disappearance. The mortality rate was staggering, concentrated especially along coastlines and in major urban centers, precisely the places where archaeologists find the most ancient remains. Teotihuacan tells a slightly different part of this story. At its peak around the 4th century AD, the city sprawled across more than 20 square kilometers in the basin of Mexico. 100,000 people or more lived there, making it one of the largest cities on the planet at the time. In 2023, Fuzuki Mizuno at Toho University led a team that finally cracked the genetic code of Teotihuacan's inhabitants, extracting complete mitochondrial genomes from 6 individuals buried in civilian dwelling areas, dated between 250 AD and 636 AD. The Teotihuacan region is brutal for DNA preservation. Volcanic ash soil, temperature swings, and humidity destroy genetic material faster than almost anywhere else in Mesoamerica. But the team succeeded, finding haplogroups A2, B2, and D1, consistent with a central Mesoamerican genetic profile. More revealing was the diversity within those 6 samples. Teotihuacan was a cosmopolitan city, pulling people from across the region. Earlier studies had already shown that neighborhoods like the Oaxacan barrio housed genetically distinct Zapotec migrants, and other barrios showed connections to Maya populations from Yucatan. The city was a genetic crossroads, a place where the diversity of pre-Columbian Mexico converged. Isotope analysis on the same Teotihuacan samples confirmed a heavy dependence on maize, the C4 plant whose large-scale cultivation had powered Mesoamerican civilization for millennia. These were not nomadic hunter-gatherers. They were urban citizens of a planned city, eating cultivated food, living in multi-family apartment compounds organized into ethnically distinct neighborhoods. And the genetic signatures they carried reflected a continent's worth of human movement funneled into 20 square kilometers of volcanic plateau. Separate research on the Oaxacan barrio within Teotihuacan, published in 2025, showed that this neighborhood maintained genetic isolation within the city itself, clustering most closely with classic period Zapotecs from northwestern Oaxaca. Meanwhile, other barrios showed connections to Maya populations from Xcaret in Yucatan. The picture that emerges is of a city where different ethnic groups lived side by side, maintaining their own burial customs and, to a significant degree, their own genetic identities, even as they participated in a shared urban culture. Each of those identities carried its own branch of the pre-Columbian genetic tree. That diversity is precisely what made the post-contact extinction so devastating. When the Spanish arrived in 1519, they did not just bring soldiers and priests. They brought smallpox, measles, mumps, syphilis, and bubonic plague. Diseases that had been circulating in Europe and Asia for centuries, building up partial immunity in those populations, hit the Americas like a biological weapon. Historical accounts describe entire villages emptied within weeks. The population that was largest, most urban, most interconnected, was hit first and hardest. the people of the coasts and river valleys, the city dwellers, the traders. The people of the Gulf of Mexico and the Pacific coast of Peru experienced decline more rapid and intense than those on the Mesoamerican plateau or the Andean highlands, as the Llamas study noted. And those coastal urban populations were exactly the kinds of people whose remains archaeologists find in caves, burial mounds, and urban excavation sites. The genetic lineages those people carried were swept away in a single catastrophic generation, or two, or three. And because those lineages had been geographically isolated from one another for millennia, there was no redundancy, no backup copies scattered across the continent. When a lineage disappeared from one region, it was gone everywhere. In 2025, a separate study added another layer to Mexico's genetic mystery. Santiago Rosas Plaza at the National Autonomous University of Mexico analyzed the gut microbiome of the Zimapan man, a naturally mummified individual from a cave in Zimapan, Hidalgo, likely belonging to the Otopam culture. About 1,000 years old, his body had been wrapped carefully in cotton cloth and maguey mats, suggesting high social status. The study sequenced bacteria from his intestinal tissue and preserved feces, revealing a microbiome that shared some families with modern humans but also contained patterns that no longer exist. Even the microbes carried by ancient Mexicans tell a story of lost diversity. So where does this leave us? The DNA from Mexico's oldest mummies and skeletons paints a picture that is both humbling and unsettling. The people who lived in these lands for thousands of years carried genetic diversity that we will never see again. Naya's lineage, haplogroup D1, persists in modified form in living populations, but the specific branch she belonged to, the version shaped by 12,000 years of isolation in the Yucatan, may be gone. The parakeet's deeply divergent genome exists only in fragments. The 92 lineages that llamas tested are extinct. The boy on the mountain carried a genetic signature shared by almost no one alive. These are not abstract data points. They represent entire communities, entire ways of being human, entire branches of our species' family tree that were cut short by forces those people never saw coming. Research continues. Ancient DNA technology improves every year, and Mexico still holds enormous untapped potential. The tropical climate that destroys DNA also preserves other kinds of evidence, bones, teeth, even gut bacteria, that new methods can now extract information from. Federico Sanchez Quinto at the International Laboratory for Human Genome Research at the National Autonomous University of Mexico has been pushing the boundaries of what is possible with degraded Mexican samples. He has compared the challenge to working with ice cream in the heat. DNA degrades faster in warm climates, making every successful extraction a small triumph against chemistry. Every new skeleton tested is another chance to map what was lost. And as the review by Roca Rada, Suilmi, Teixeira, and Llamas in 2020 made clear, Mesoamerican ancient DNA research is still in its early stages. Despite a rich archaeological record and indigenous populations that descend from some of the most famous civilizations in human history, Southern North America and Central America remain among the least genetically studied regions on the planet. The Olmec, the oldest known civilization in Mesoamerica, only recently yielded DNA. Two burials from San Lorenzo Tenochtitlan, one dating to 1200 BC and another to 1000 BC, were sampled by Enrique Villamar Becerril under the direction of Anne Cyphers at the Institute of Anthropological Research at the National Autonomous University of Mexico. Both individuals carried haplogroup A, one of the most abundant founding lineages in the Americas. That confirmed the Olmec as genetically indigenous American, settling a long-running debate about their origins, but it also raised questions about what other haplogroups existed in Olmec society that we simply have not sampled yet. The oldest mummies in Mexico carry DNA that does not match any living human. That is not a mystery waiting to be solved. It is a record of what happens when entire populations, isolated and unique and irreplaceable, collide with a force that does not care about their uniqueness. The genetic silence in those bones is not empty. It is an echo of a world that was here long before ours, and that vanished faster than anyone thought possible. Every ancient genome we recover is a small act of restoration, pulling a lost voice back from the dark, even if only as data on a screen. The bones have been waiting. The technology is finally catching up.